Hi guys, welcome to our first asynchronous discussion video. Today I'm going to be talking about enzyme kinetics. I'm going to do a quick overview of stuff that we talked about in discussion in the live version before going into the worksheet answers. So first thing I wanted to talk about is the induced fit versus lock and key model of enzymes. This is something that I am just kind of passionate about because I am a huge proponent of mechanistic relationships, finding out why something is occurring instead of just knowing that something occurs. So the lock and key model is kind of an outdated, more elementary version of how enzymes work that essentially just says, okay, you have a substrate and you have an enzyme and they fit together, and then magically these products appear and the enzyme is ready and rearing to go for the next one. The induced fit model is a little bit more in-depth than that, which I prefer. It shows that you have this substrate and you have an enzyme with an active site, but when they fit together, they form this thing called the enzyme substrate complex, which changes the shape of both of those things a little bit. So both of those are kind of adjusted based on their fit together. And something about the way that it changes shape really causes the enzyme structure to allow for the product formation. So something is happening on a molecular level that's causing that reaction to occur at a way faster of a time scale than if it was just to occur on its own. So this could be bending it so that certain bonds break, or putting certain charges closer together. We don't really know what exactly, it's going to depend on each enzyme, what is taking place that's going to cause this to happen. But we do know that something happens to the shape that favors the product formation. So that's how it works. And then once the products are released, the enzyme does go back to its original shape unless we denature the enzyme, which I'll talk about in a little bit. But that's why enzymes are so great, is because they return to its original shape. So then suddenly you can catalyze more and more and more, and you don't have to replace them. So hopefully that makes a little bit of sense regarding structure. One other thing I wanted to cover real quick is about this graph. So I feel like a lot of people have trouble at first when they're hearing about enzymatic reactions. They have trouble picturing why we need activation energy at all in this case, because we look at this graph and we see that, okay, this is an exergonic reaction overall. You're releasing energy. It seems like we should just be able to go from the reactants to products and energy is released and all is dandy. But the thing is that anytime you're making a significant structural change, it's going to require a little bit of momentum to or a little bit of work to get that to happen. It doesn't matter if the change is ultimately going to release energy. It's just you need that initial input in order to initiate that original change. So that's going to form something called an intermediate. It's like an intermediate structure that's the shape that the enzyme substrate complex forms that makes it advantageous for the products to form. And that is the part where forming that intermediate step is the part that requires activation energy. So if we compare the no enzyme reaction versus the enzyme reaction, we see that the reason why enzymes can speed up so fast is because making that intermediate structure is going to be a lot easier when the shape is already almost there for you. It's essentially you're aiding it along, so that you don't have to wait for it to occur naturally. You're kind of you're giving it a little bit of that extra push to get it going. And so reducing that amount of activation energy that's needed is going to cause the reaction rate to increase by a lot, usually. And yeah, so the difference in rates between the regular spontaneous reaction, how it just occurs in nature, versus how fast the enzyme will go, will depend on the difference between the activation energy between the two. If you have a huge reduction in activation energy, you're going to see a big increase in reaction rates when you add the enzyme. If you barely improve the activation energy, you might not see a huge difference between enzyme and no enzyme. All right. Okay, so some key terms for doing the worksheet and just for learning about enzymes in general. So Vmax is a really important thing to know. It's the maximum speed of the reaction. V naught, or V sub 0 is what it looks like, but it's pronounced V naught, is like the initial speed. So like when we first started the reaction, what was the initial speed? Km is the concentration of substrate that's required to reach half of the Vmax. So at first glance, it kind of looks like this is such a random measure. Like it just seems totally out of the blue. The reason why we use it is because it's essentially a proxy for measuring affinity, affinity of the substrate to the enzyme. So if we think about it, if we have a really high Km, what that's telling us is that we need a ton of substrate in order to get to just half the Vmax, which isn't that high. So it probably means that, okay, like if you need a ton of it just to get to half, you're probably not having like a great fit between the substrate and the enzyme. If you have a really, really small Km, that means like you need just barely any substrate in order to get up to that Vmax, which indicates really high affinity. So that's kind of why we use that measure. I know that it seems weird at first glance. And just one thing to remember is that when you're asked to calculate the Km, really be careful that you're not just calculating the half Vmax point, because half Vmax is just the rate at like the half maximum rate. We need to find the substrate concentration that will get us there, and that will tell us the Km. So just be careful of that, because that's like a hugely common mistake that I see. Next big thing is inhibitors. So competitive versus non-competitive are our two main types. Competitive inhibition is essentially, it's kind of self-explanatory, but you're competing directly with the inhibitor for the active site. So you have all these enzymes, they're ready to go. They can't really differentiate between what's what. So they don't care if it's an enzyme or if it's a substrate or if it's an inhibitor that's coming in, they're going to process it regardless. So the interesting thing with that is that it's essentially just a numbers game. It's just like chance. Are you getting the active, or are you getting the substrate or the inhibitor in there? And so in order to like mitigate the effects of inhibitors is that you can just flood the solution with more substrate. And so like essentially the higher ratio you have of like the substrate to inhibitor, the closer to the original Vmax you're going to get, because it just like probability will just say, okay, you're going to get so much substrate in there that it's like the inhibitor is not going to even really do anything. It's going to be negligible. So you're just playing a numbers game. With non-competitive, it's kind of the opposite. So non-competitive, the inhibitor is binding to somewhere else on the enzyme, but it's causing some sort of structural change that changes the active site enough that it can't bind to the substrate any longer. And in that case, you can keep adding substrate all you want, but if the active site is of no use, then you're not going to be able to get anything. You're not going to be able to get a reaction out of that regardless. So that's kind of like the big non-competitive. The Vmax is going to be very different, because when you add the inhibitor, you're kind of like altering the shape of the active site in general, regardless of how much substrate is in there. So your Vmax is going to go down no matter what. Km, the pattern is harder to tell because it's so specific to each like specific structure, like what structural change is the inhibitor causing to the active site. That's going to tell you how much substrate you need to get to half the Vmax, and you can never predict that. You can calculate it if you're given the right information, but you can't predict what's going to happen to the Km because it's just so specific to each thing. So hopefully that makes sense. We're going to look at it more in depth with the worksheet, so don't worry about that if you're a little bit confused still. Cofactors is another thing that you might hear about when learning about enzymes that I think is really important. Cofactors refer to any non-protein chemical compound required for the reaction to occur. So we remember that enzymes are proteins, right? So anything that's not actually part of the enzyme that is required for it to go can count. The main ones are ions and organic molecules. Iron is a really common cofactor. Copper is a really common cofactor. There's some other really common ones. And then the word coenzyme is kind of a subset of cofactors. So any organic molecule cofactors are actually referred to as coenzymes. And you'll see that a lot in the literature and stuff. So I don't want you guys to get confused by that, but it's literally just a type of cofactor. Examples of these are B vitamins, flavins, you'll actually see in multivitamins and stuff a lot of coenzymes. So it's just like they help reactions in your body. Cool. So another thing I wanted to mention is, yeah, enzymes can only speed up reactions that would occur naturally in nature. So if you add an enzyme to two things that would not normally react with each other, you're not going to suddenly create a reaction. The only thing that the enzyme is doing is reducing that activation energy hump to get there. So yeah, so essentially like anything that can happen in nature can be sped up by an enzyme if it like sufficiently decreases that activation energy hump and push that reaction along. That's all I wanted to say about that. And generally, enzymes are reusable. We will talk about denaturing enzymes, but that's the cool thing about having enzymes that can be reused. It's very efficient. Okay, so if you haven't done the worksheet already, hopefully you have at this point, but if you haven't, what I would do is get a sheet of paper out and kind of write down some like just really quick predictions about what you think is going to happen to the graph of the reaction rates based on these different factors. So what we're going to be looking at for the worksheet is temperature, pH, substrate concentration, and then the two types of inhibitors, the competitive and non-competitive. So I would just do like a really quick, like, okay, what is the shape of the graph going to be like? And then maybe like a quick note about like why you think that is. Doing that is going to really help identify where you are struggling with like learning about enzymes versus like what you already know down pat. And when you compare them to the answers, you'll be able to kind of see like, yeah, like you'll be able to kind of see the flaws in your thinking or just like better identify errors and stuff that way. So I'm just a huge fan of doing that. So before we actually like look at the graphs in the slides, I wanted to just mention the thing with enzymes is that there's so many different things that you can graph that are useful for us. So when you're looking at a graph of like something related to an enzyme, be really, really, really careful that you're looking at what are the axes representing, like what are their units, what are they measuring, what are the individual data points represent, and why do we care? Like what are we trying to glean from the graph that we're given or that we plot ourselves. The reason why this is important is because, so like here, this is a screenshot from the worksheet that we've been given. We can see we have temperature on the x-axis, so that's the conditions that we're comparing. And then we have the reaction rate on the y-axis. So these are the V naughts. And the important thing about that is if we have a graph that looks like this, the red line, like what's going to be happening at the red line? If this is a rate, when we see a plateau of a rate, a lot of people in my like previous 173 classes and stuff would be under the misconception that suddenly the reaction is stopping here and that no more product is being formed because we see a plateau, and so you might think like, okay, we're done. But no, because this is a rate. So this means that the product is still being formed and stuff is still happening. It's just that the rate is no longer accelerating. It's no longer increasing. So we've reached Vmax at this point, right when we get to the plateau. That's Vmax, but you're still making product. And so we could also be given graphs of product formation. If we were given graphs of product formation, then this plateau would represent the point where the reaction stops. So either you run out of substrate or whatever. Yeah, so hopefully that makes sense about why you have to be like just really careful. And also just recognizing that like in this case, we're looking at the initial rates of different reactions at different temperatures. In other cases, we could be measuring like the reaction rates of one reaction over time. So we could be like, okay, let's do this at 30 degrees, all at 30 degrees, but we're gonna measure at each minute and see how long it takes to reach Vmax. You can look at the product formation, you can look at the different conditions. There's so many things that you can look at that being really careful is just like super, super, super key. So just want you to remember that. So now hopefully at this point you can pause and go through the worksheet, try to do it on your own as best as you can, try to make predictions, try to think, okay, what mechanism is happening here that's causing this, whatever. And then I'm going to switch my screen over to the other PowerPoint so we can discuss the answers. Okay, so here's the slides. Hopefully you can read these big paragraphs because I'm not going to go over them, but they are useful. So experiment 1, we have temperature here. Hopefully you guys got a graph that looks kind of like this. If you didn't, you probably just made an error in your Excel or something like that. You should all get a graph that looks like this. Make sure that your scale is good enough so that you can actually kind of see the different data points. So we look at the graph, we can see, okay, reaction rate, temperature, cool. We're looking for the optimal temperature. And remember, we said that typically we consider the Vmax to be the optimal because we generally want enzymes to be as efficient as possible. So that's why we consider highest speeds to be optimal. We can see that this occurs at around 50 degrees Celsius because it's about this point in time. And it's interesting because after this point, we see it goes back down and we're wondering what is happening at this point. We might have expected this increase here. And if you did, that's good because we do. As the temperature increases, the molecules are, buzzing about more and more. And you just, again, it's a probabilities game. The faster you move, the more likely you're going to hit an active site and be able to form a complex. So it's literally just like increasing the chances of making that happen. And that keeps on happening until we get to this point where suddenly any higher temperature is going to cause structural changes that are going to affect the active site and its ability to bind with the substrate. So if we remember from last week, we talked about what kind of things cause this at high temperatures, what's happening here. So try to think about that. But it really is just the breaking of bonds. So bonds are being broken, things are being cleaved, and shapes are changing. And once you get to a point where the shape is just like so far gone, you're gonna essentially get no enzyme activity left. So that's why we kind of see this gradation. It's not like a quick drop off, like suddenly right past 55, you like get no function. It's essentially like, okay, so like one bond is broken and you can like kind of fit the substrate, but it's not as ideal. Like you still have a little bit more activation energy to get over that kind of thing. And so yeah, that's why we kind of see like a gradual decrease. So hopefully that makes sense. One thing that's interesting to note is that even if you bring the enzyme back to its optimal temperature, it's still going to be denatured. So this is like a permanent thing. Like once you get up to these really high temps, then you're not going to be able to go back. There are some enzymes that have really advanced like repair mechanisms that can repair covalent bonds and whatever, but that's like not the norm at all. So just assume that if you have a denatured enzyme from temperature, it's staying that way. I'm going to post this. I'm not going to read these paragraphs, but it's essentially just what I just talked about. Heat-resistant bacteria, we talk about extremophiles, right? So we wanted to like guess what would happen to this graph. Hopefully you made a prediction. But we would expect to see is that the graph would just be shifted upwards on the x-axis. So we would see that the optimal temperature is at a much higher temperature than a typical enzyme. But we're still going to see that same shape. So the pattern itself is not any different. You're still going to see that, okay, once you are approaching and approaching and approaching the optimal temperature, you're getting faster and faster and faster because of those collisions. But then suddenly at a certain point, even the extremophiles are going to start breaking and having those shape changes that cause the reaction to not work anymore. So yeah, hopefully that makes sense. These are the answers. I think I covered all of these. Yeah, I did. Okay. Experiment 2, pH. Okay, so this one is a really important one, and it's one that can be tricky, especially like without hearing any explanation. If you just look at the slides, it can be tricky. So important thing to note, first of all, is the background. Okay, where is invertase coming from? If we look at slide 9, we can see, okay, it's a membrane-bound enzyme whose active site projects out into the lumen of the small intestine. So we know that small intestine, stomach, all that, like digestive tract stuff is usually highly acidic. So you can kind of predict, okay, if it's functioning there and it's, co-evolving with us, it probably is going to function pretty well in acidic environments. So that's just kind of like our baseline prediction. We look at this first graph and we see, okay, cool. So we see that around a little bit below 4, a little bit above 4, that's kind of a range where it's functioning pretty well. Its Vmax is like right around pH 4.5. But then we see these interesting trends of like, okay, it's not functioning very well below like 3 or above like 5. So we have this really narrow range in which it works. And you might be tempted to say, okay, cool. So now we know that enzymes function best in this range. But the thing is that this is so dependent on the structure of the enzyme and like whatever substrate that you're catalyzing, that pattern is specific like for this enzyme, but it can't be extrapolated to other enzymes. So essentially what we want you to get out of this is that you're going to have a range, an optimal range. We don't know what that range is per enzyme. Like if I just gave you a random enzyme name and no background about it, you wouldn't be able to predict where, like what that range would be or how big that range would be. But it will have an optimal range. And the reason why that is because if we have that particular structure in place that somehow like allows the products to be formed like pretty easily, if we make lots of changes, so like if we're adding a ton of protons in, we're like ionizing things, like changing acidity, eventually that structure is going to start to change because of that in either direction, whether you're deprotonating, or adding a bunch. It doesn't matter. Eventually the shape is going to change because of that. And once you reach that point where the active site changes, that's when the enzyme stops functioning. So some enzymes might have a broader range than others, depending on how, just like the structure of it, it's so complex, but it's just like, okay, how much deprotonation and protonation can you handle without changing the active site? So the range might be even like a different size based on enzyme, but also like what the actual range is will differ. And it's literally just because of these structural changes from adding and taking away these protons. So just remember that like if you memorize these slides and are like, okay, 4.5 is the optimal pH, that's like probably not going to be true for a lot of the enzymes that you study. So just remember that it's not, this isn't a pattern that you can extrapolate out. Also for, I don't think I specifically went over question 1 and the temperature one either, but all the question 1s on this worksheet is really just to help you guys think about, okay, why do we need to control for all of these variables? And hopefully that should be obvious now, but if we alter the substrate concentration at the same time as we're altering the pH, for example, we're not going to be able to know what change in the data points are attributed to which variable. That's kind of just like a scientific like mode of thinking. Like we need to be able to attribute the change to a single cause. So we have to control for everything else. So that's kind of why they're asking it again and again. For number 2 for this, so where do you think this enzyme's pH optimum will lie? The reason why we're able to provide an answer is because we're given that context about its location in the human body. If this was an exam and I just gave you a name of an enzyme, you wouldn't be able to accurately predict that most likely. So yeah, the context does matter. Optimal pH, yep, that's just the pH at which you reach the Vmax, which we could see from here. So we see, okay, Vmax is reached here, so we'll look and we'll see what pH it's at, about 4.5. And then I already kind of explained how pH changes affect it. You can look back at the discussion slides from last week to see what protonation and deprotonation does to protein structure. And it's like the same theory. Okay, experiment 3, substrate concentration. Okay, so here what we have is a graph where we see, okay, we're adding a bunch of substrate, adding a bunch of substrate, adding a bunch of substrate, and then suddenly we get to this point where adding more substrate doesn't do anything else. And so we think about this and we're thinking, okay, so what happens at that point which will cause the reaction to stall, even if we're still doing the same thing that we were doing before, adding more substrate. And hopefully you can predict what that is, but I'll tell you anyway. It's when the enzymes become saturated. So enzymes are full, they don't have any extra capacity, they don't know, they're not aware of how many are waiting in line for it, they don't know that. So they're just going along, and you can add as much substrate as you want at that point, but they're still going to go at the same rate because they're full. So there's nothing else that you can do. So that's what happens when you reach that plateau. Now, if you wanted to counteract that plateau, one of the things, if you didn't look at the slide already, is hopefully pretty obvious that you can increase the amount of enzyme that you have. If you have a ton of substrate available, there's nothing limiting that. So you just add in more enzyme, and then suddenly your reaction is going to pick right back up again because you have more capacity to filter through those. Additionally, what you could do is you could just change the pH or the temperature conditions to their optimal positions for that specific enzyme. And the reason why this is really important to note is that a lot of times in like lab settings, research settings, medicine, stuff like that, we just don't have a ton of enzymes lying around, especially if we're like researching extremophiles and like things that are rare. We can't just like add a bunch of enzymes because we want to. So knowing this is really helpful, because if we're limited of enzymes, we can say, okay, what's the pH that it works best at? What's the temperature that it works best at? Let's do that, because then automatically we're going to get a bump in that Vmax without having to even change the amount of enzymes available, which is really cool. Okay. And then hopefully you were able to calculate the Km. So check yourself here, especially. See that when you calculated Km, you didn't accidentally say, okay, the Km was 0.0015, because that's just half the Vmax. Whereas Km is saying how much substrate did you need to get there. So the Km in this case, go back down to the x-axis, and this looks like around 25 millimolar. That's what's getting you to the half Vmax. So just make sure that you didn't make that mistake of accidentally doing the volume instead of the concentration. I believe we talked about all of that. Yes, we did. Okay, skipping over these, but make sure to read them. They are important. So now we're doing the inhibitors and trying to compare or trying to guess which one is competitive or which is non-competitive. So let's look at this one first, experiment 4. You can see here, there's actually really not that big of a difference between them, right? There's like a negligible difference in Vmax, I would say. And the Km, so if we look, like the Km is going to be around 0.0015 for both of these, because they're both around 0.003. So we do see like this tiny little difference in Km. It does require a little bit more substrate to get to half the Vmax, and the Vmax is like technically a little bit lower for the inhibited one, but these are so close that they're essentially the same. This probably indicates to us, hopefully you said competitive inhibitor. And the reason why is because adding a little bit more like substrate is going to actually help this retain almost exactly the same Vmax. So we just add a little bit more, and we're not getting totally up to the regular Vmax, but we're pretty dang close. And so the reason why that is competitive is because, remember, if we want to keep the same Vmax with competitive inhibition, we can just flood with more substrate, and then like the rules of chance will just dictate, okay, cool, then that's the amount of substrate that will be processed. So we add more substrate, and then we retain about the same Vmax. So that's a good indicator of competitive inhibition. You can read these little paragraphs for more information on that. And then looking at this one, so here we can obviously see between the inhibited one and the non-inhibited one, there's a huge reduction in Vmax when you add the inhibitor. So it's like almost half. It's like about half the Vmax. And so what that's telling us is that in this reaction, we can add as much substrate as we want. Like, look at how much substrate we're adding, and essentially nothing is changing. So that tells us, okay, something is happening with the enzyme itself. So like something has changed on the enzyme itself to make those active sites ineffective. And that tells us non-competitive inhibition, right? Because something has bound to it and caused the shape to change. And that way you can add as much substrate as you want, nothing's going to be processed through that. So that's how we can kind of dictate, okay, this one is a non-competitive. Now the interesting thing with non-competitive inhibition is that the Km patterns are going to be very like like not proportional to what you might think. So we generally ignore KM for non-competitive, and I'll kind of explain a little bit why. Here we can kind of see essentially the exact same KM values, whereas we have like double the Vmax. And this is only in this particular case. We could have some cases where the KM is a lot different. And really that just depends on these really complex, like subtle changes that determine like what is the inhibitor doing to the enzyme? Like what structural change is it making? And how does that affect how much substrate we need to add in order for this reaction to occur at like a relatively like normal pace? We can never predict that, at least with like the amount of information that we would give you in this course. So you'd never be asked to predict what type of competitor is this, like based on just the KM alone. That being said, you can still calculate the KM when you're given the Vmax value. So when you're given a chart like this, you can still calculate what the KMs are and be like, oh, hey, in this case, they're practically the same. But you would never be asked to predict the difference in KM for a non-competitive inhibitor. So hopefully that makes sense why. It's just because the unique, tiny little changes will affect what pattern that has. So it's kind of like the pH effect, where it's going to be so independent, or it's going to be so like unique to each particular thing that we're looking at. You can't like make a universal statement about what's happening there. So hopefully that makes sense. I think I talked about all of these questions. Yeah, so just being able to look at Vmax and KM on a graph, being able to either calculate them or interpret them. So like, what does it mean when we see that the Vmax has been halved when we add an inhibitor? What does that tell us? Being able to, yeah, kind of predict what type of inhibitor we have. Being able to just explain what's happening at each of these steps. So like when we see a plateau or when we see a peak and like something like is increasing and then decreasing, like an inflection point, what is happening at those stages? That's what I really want you guys to focus on, because I don't care if you remember, like you can memorize all you want, like, okay, competitive, Vmax will stay the same, KM increase, non-competitive, Vmax decreases. Like you can remember all the patterns that you want for cramming for the exam, but that's not really going to help you. And this, I can promise you, this comes back so much in upper level science classes. So you will need to remember these patterns, and to really remember them, it's just easier to know why, like what is happening on this like molecular structural level that's causing these patterns to exist. So I highly suggest just going through these concepts in your head, making sure that they make sense to you, not just that you can memorize them, just because in the long run that will be a lot better, and it'll, if you have a brain freeze during an exam, this will be a better way to learn. Cool. So that's basically all I have. If you go through the quiz questions, you should be able to tell that a lot of the themes from the quiz questions are kind of reflected in here. So hopefully that answers a lot of the questions that you have about that. If you do still have quiz questions for me, please feel free to reach out. I am happy to answer any. And overall, thank you guys for being willing to kind of go on this weird adventure of finding out which structure to use. I'm excited to try this and hope that it works. Again, if there's feedback, I'm totally open to continuous feedback. I'm willing to change things and improve them so that everyone gets the most that they can out of this class. So yeah, please keep in touch. And I did send out the office hours link. And I'm going to be inviting Google Calendar invites to all of you guys for that. So please come to my office hours or to Sarah's or to Lori's. And we are happy to answer any questions that you may have. All right, have a good one.